Hello everyone, welcome to EduHelpDOTCOM. In today's video, we are going to learn recombinant DNA technology. This is a very important 15-mark question for your exams. Let's make it simple and easy to remember. Recombinant DNA technology is the process of combining DNA from two different organisms. It is also called genetic engineering. This technology is widely used in medicine, agriculture, and industries. The first step is isolation of DNA. DNA is extracted from the donor organism. The cell is broken using enzymes like lysozyme or cellulase. RNA is removed using RNase. Proteins are removed using protease. Finally, we get pure DNA. Next step is restriction digestion. Here, restriction enzymes cut the DNA. Both donor DNA and vector DNA are cut using the same enzyme. This ensures compatibility for joining later. Now, DNA fragments are separated using gel electrophoresis. From these fragments, the desired gene is identified and isolated. The selected gene is amplified using PCR. This means we make multiple copies of the gene. It increases the quantity of DNA for further steps. In this step, the gene is inserted into a vector. The enzyme DNA ligase helps in joining them. This forms the recombinant DNA molecule. Now, the recombinant DNA is introduced into a host cell. This process is called transformation. It can be done using heat shock or electroporation. Not all cells take up the DNA, so we select only the transformed cells. Marker genes help in identifying these cells. The selected cells multiply and express the gene. As a result, the desired protein is produced. For example, insulin. Finally, the product is extracted and purified. This is called downstream processing. The final product is then used commercially. Now let's understand restriction enzymes. They are called molecular scissors. They cut DNA at specific sites. There are two types of cuts: sticky ends and blunt ends. Sticky ends have overhangs and join easily. Blunt ends are straight cuts and are harder to join. Restriction enzymes ensure precise DNA cutting. They create complementary ends for ligation. They are essential for gene cloning. Here is a quick trick to remember all steps. I cut, isolate, amplify, ligate, transfer, select, express. Try repeating this once or twice. Recombinant DNA technology is a powerful tool in biotechnology. Restriction enzymes play a key role in this process. They make gene manipulation possible. If you found this video helpful, don't forget to like, share, and subscribe. Follow EduHelpDOTCOM for more exam-focused content. See you in the next video. Thank you.